Alright, today we're going to be going over the heart lab because obviously we've been snowed out again. So we can find this in the beginning of the heart chapter, or the heart PowerPoint. And so first we'll talk about location and position of the heart. The heart's enclosed in the pericardium within the thoracic cavity, which lives within the mediastinum. So it sits posterior to the sternum, a little bit left of body midline, about 2/3 of it is to the left. It sits in between the lungs and the mediastinum. It is slightly rotated so that the right side is more anterior than the left. We have the base, which is going to be, I know it makes little sense, but the base is kind of on top, the posterior anterior side, and then the apex right here is going to be more inferior. So as you can see, this is sort of where it is located in the mediastinum in between the two lungs. And we have the right atrium up here, the left atrium, and the right ventricle is much more easier to see, and then the left ventricle is a little bit further back. So first we'll talk about the pericardium. These are three-layered, the fibroserous sac around the heart. And we talked about this in 168 a little bit. Remember we had the visceral and parietal serous membranes, so we're kind of going back to that. But we'll talk, there are three layers of the pericardium you guys will be responsible for. The fibrous pericardium is the outermost covering. This is made out of dense, irregular connective tissue, and it attaches to the diaphragm, the base of the aorta, and the pulmonary trunk, and it anchors the heart and prevents it from overfilling, right? So this is like tough and fibrous material. Then next, as we continue moving inward, is the parietal layer of the serous pericardium. This is the simple squamous epithelium and areolar connective tissue. We talked about body membranes a little bit in 168. And this is attaches to the inside of the fibrous pericardium. And I'll show you an image in just a moment. Then the visceral layer of the serous pericardium. This is the part that directly covers the heart. And it's once again simple squamous and areolar connective tissue that attaches directly to the heart. And the two serosal layers are continuous and separated by that pericardial cavity, which is filled with that serous pericardial fluid. And so in this picture we can see the fibrous sac is on the outside. Then we have the parietal layer of the serous pericardium, right? So the fibrous sac kind of makes up the pericardial, like the wall of the pericardium. Then the parietal layer lines that wall. Then we have our parietal cavity that contains the serous fluid. And then on the inside is the visceral layer of the serous pericardium. And this can also be called the epicardium. And we'll talk about that when we talk about the layers of the heart. So there are some important superficial features of the heart you'll need to know. There are some really good videos that I linked that go over some of the superficial features of the heart on models. You guys should watch those. But the first thing we'll talk about are the grooves that mark the borders of the heart chambers, and we can see these from the outside. And we call those sulcuses or sulci. First, we have the coronary sulcus, which separates the atria from the ventricles, and this extends around the circumference of the heart on the top. So we can see here, we're going to have the coronary sulcus is going to go around here, and that's kind of between the atria and the ventricles. Next, we have the interventricular sulci, and they separate the right and left ventricles, and we'll be able to see one on the anterior side, the anterior interventricular sulcus, and one on the posterior side, the posterior interventricular sulcus, and they go from the coronary sulcus towards the heart apex. And these grooves are going to contain the coronary vessels that supply blood to the heart, and we'll talk about those in a bit too. So you can kind of see, this is the anterior surface, so we can see this right here is going to be the anterior interventricular sulcus, and the posterior one would be on the posterior side. All right, so on the anterior view, other things we can see are the right atrium and the right ventricle. They're very prominent. We can see those very easily. The right auricle is a wrinkled extension of the atrium. It kind of looks like the ear of the atrium and is especially noticeable. We can see the left auricle over the left atrium, but it's not as easy to see. The aorta and pulmonary trunk, and then some of the anterior interventricular sulcus can be seen, right? So we have, this is the aorta right here, the aortic arch. This is the pulmonary trunk right here, right? So we have our pulmonary arteries. Then we have our right atrium, and the right auricle is this wrinkled part here, and the right ventricle. So we can see a little bit of the left ventricle and the left auricle over here. So on the posterior surface of the heart, we can see the left atrium and the left ventricle are more prominent. So we can see the left stuff a little bit better on the posterior view. The left atrium forms the base on the posterior superior surface, right? So remember, the base is superior. Also visible are the pulmonary veins attached to the left atrium. We can also see the superior and inferior vena cava and the pulmonary arteries well from this view, as well as the posterior interventricular sulcus and part of the coronary sulcus on the back that houses the coronary sinus. So here we can see the aortic arch coming down, and this is going to become the descending aorta. The top of the left pulmonary and right pulmonary arteries, they branch off from that pulmonary trunk. This is going to be the left atrium. This is kind of the back of the left atrium that makes up the base, and we can see the left and right pulmonary veins entering that left atrium. This is going to be the coronary sulcus, and this big blue vein here looking thing is the coronary sinus. We can see the superior vena cava and the inferior vena cava where they enter the right atrium. Down here we can see the posterior interventricular sulcus with its coronary arteries and veins. The left ventricle is most prominent, and a little bit of the right ventricle is here. This bottom part is the apex of the heart. So now we'll talk about the layers of the heart wall, and this will also be important. Make sure that you can look at this diagram and know what that is for the quiz. So the heart wall is going to vary in thickness. Your book has a really good picture that I don't have in this lecture, but it compares the thickness of the right and the left heart wall. Which do you think is thicker? So if you remember, since the left side of the heart is going to be pumping blood to the whole body, it's going to be thicker. So the left ventricle has a thicker wall than the right ventricle. And in general, the ventricles, or the pumping chambers, have thicker walls than the atria. The heart wall has 3 layers. The outside is going to be the epicardium, and so this is also the visceral layer of the serous pericardium. So the epicardium is synonymous with the visceral layer of the serous pericardium, and so it's made of the simple squamous on top, overlying the areolar connective tissue. The middle layer is the myocardium, right? Myo, remember we learned, means muscle, and cardium is heart, so it's the cardiac muscle. And this is going to be like the pumping area. And then the endocardium is going to be a thin layer of simple squamous overlying areolar connective tissue that lines the inner cavities of the heart. The endocardium is also important that it is continuous with the lining of the blood vessels. So it's going to line the inner portion of the heart, and then it's going to continue to follow like the arteries and the vessels. So remember we talked about our heart having 4 chambers. We have some separation of the chambers internally. They're all separate chambers. There's no mixing of oxygenated and deoxygenated blood. The interatrial septum separates the left atrium from the right atrium, and the interventricular septum separates the left ventricle from the right ventricle. Make sure you can find those and identify them. We'll talk about the atria first. We have a right atrium and a left atrium. They have some ridges on the anterior wall and within the auricle, and we call those are the pectinate muscles. We can find the fossa ovalis, which is an oval depression that's right in the interatrial septum. And this is going to be where the fetal foramen ovale was. So when the lungs were not functional, blood was shunted from one side to the other, from the right to the left atrium during fetal life. And then once the baby wakes up and takes their first breath, this is going to close, and that is going to turn from the open foramen ovale into the sort of closed fossa ovalis, which is kind of like a scar in the interatrial septum. The right ventricle receives blood from the body via the vena cava and the coronary sinus. The left ventricle receives blood from the pulmonary veins, and the blood is going to exit both of those through the atrioventricular valves. And we'll talk more about the valves in a bit. Next, we have the ventricles. They're going to be the lower chambers, right? They're thicker walls. There are some muscular ridges inside the ventricles that you'll be able to see on some of the models. We call those the trabeculae carneae. And then you can also see some papillary muscles. So papillary muscles are big cone-shaped projections that extend from the ventricle wall. The right ventricle has at least 3 of them, the left has at least 2, and they're going to anchor the chordae tendineae. So they are going to be attached to the chordae tendineae. The chordae tendineae, or heart strings, or tendinous cords, are thin strands of collagen that attach to the atrioventricular valves. So we have the papillary muscles that attach to the chordae tendineae, and then the chordae tendineae attach to the leaflets of the AV valve. And you guys, if you watch the model video, you can see those, and we'll have some pictures too. The right ventricle, blood is going to exit through the pulmonary trunk to the lungs, and the left ventricle, blood exits through the aorta to the body. All right, so we can see We've got the right atrium up here. This is going to be the opening for the coronary sinus. This is the fossa ovalis, so that scar where the foramen ovale used to be. And then we have the entrances you can see from the superior and inferior vena cava. The pectinate muscle is seen here. This is going to be one of the AV valves, and this is the other. When we move down to the ventricles, we can see these guys here are the papillary muscles, and the papillary muscles are going to be attached to the chordae tendineae. These are these little cords, and the chordae tendineae are in turn attached up to the leaflets of the AV valves. And then we can also see these trabeculae carnae, which are those little ridges inside the ventricles. Then we have our semilunar valves, and those are the valves that are going to divide the ventricles from the great arteries that are leaving the heart. The heart also has a fibrous skeleton of dense irregular connective tissue, and this is going to be sort of, it provides a structural support, and this is at the boundary between the atria and the ventricles. And so we can see this white part here is going to be that fibrous skeleton kind of between the atria and the ventricles. And so this is important because it provides a framework for attachment of cardiac muscle. It also forms fibrous rings to anchor those valves, and it acts as an electrical insulator that prevents the ventricles from contracting at the same time as the atria. So this will be really important when we talk about the cardiac cycle and the electrical activity of the heart. So the heart valves are really important because they ensure one-way blood flow. We only want the blood to flow in one direction. If valves become leaky or too stiff, or they don't close properly, we could have blood flowing in the opposite direction, and that is not efficient. It's not very good. We have two types of heart valves, the atrioventricular, which are here in green, and they're between the atrium and the ventricles, and then the semilunar valves, which are shown in yellow, and they're between the ventricles and the great vessels. All of them are made of endothelium-lined fibrous connective tissue cusps or flaps. And so the atrioventricular valves here prevent backflow into the atria. So the blood will flow through the AV valves from the atrium into the ventricle, and then when the ventricle starts to contract, the valves are going to snap shut. And so when the valves are closed, you can see that the blood in the ventricle sort of pushes against them, but the papillary muscles and the chordae tendineae are going to keep them closed well, so that they don't, the blood is not allowed to go back into the atrium. And so one valve is in each atrioventricular opening. So the right AV valve is called the tricuspid valve because it has 3 flaps. The left AV valve is called the mitral valve or bicuspid because it has 2 flaps. Then we have the semilunar valves, and the semilunar valves prevent backflow into the ventricles. And so they're open when the ventricles contract and blood flow goes through them into the vessels, and then they're closed when the ventricles relax. When the ventricles relax, the arterial pressure is going to be greater than the ventricular pressure, right? So the blood is going to want to flow backward, but these semilunar valves are going to snap shut and stay closed, so the blood cannot go back into the heart and stays into the vessels. Each of the 2 semilunar valves has 3 cusps. The pulmonary semilunar valve in the right is between the right ventricle and the pulmonary artery, and then the aortic semilunar valve on the left is between the left ventricle and the aorta. You have a link in the lab folder that is going to let you listen to some normal heart sounds. The 2 familiar heart sounds that we know are lub-dub, and those are S1 and S2, and we can sort of see here this is like a waveform of what it would look like. We have S1, the lub, and then the dub. And these are, the sounds happen because of closing of the valves. And so S1 is when the AV valves close, and that happens a little bit before S2, which is when the semilunar valves close. There are 2 minor sounds, S3 and S4, which are not often heard, and they may indicate disease processes. But we're just going to focus on S1 and S2, which are the normal sort of lub-dub. And then each heart valve can be heard best at specific anatomic locations, right closer to those if you're closer to the heart valve. So the next thing we'll talk about are coronary vessels, and there's another video that you can watch to look at the coronary vessels. They are blood supply within the heart wall, and the coronary circulation delivers blood to the heart's myocardium, and that's really important. They transport oxygenate the arteries transport oxygenated blood to the heart wall, and the veins transport deoxygenated blood away from the heart wall and towards the right atrium. And the heart feeds itself first, so it's going to be one of the first little branches off of the aorta. And so our coronary blood flow is intermittent with the heart contractions. The vessels are patent or open when the heart is relaxed, and the vessels are compressed or the flow is interrupted when the heart contracts. So we have a couple of coronary arteries and veins that you guys are going to have to be able to identify in your models. The right and left coronary arteries are going to sit in that coronary sulcus that we talked about. They're the first and only branches off of the ascending aorta, right? The heart feeds itself first. It's really important that it has its own blood supply. So the right coronary artery is going to branch into two branches, the right marginal artery and then the posterior interventricular artery. And then the left coronary artery branches into the circumflex artery and the anterior interventricular artery. And so this one, the anterior interventricular artery, is going to be important. It supplies the anterior surface of both ventricles and most of the interventricular septum, but it's also sometimes called the widowmaker nickname because there's a high risk of fatality if that one becomes occluded. So we can see here our coronary arteries. We have our right coronary artery, and the branches are going to be to the right marginal artery, which is here, and then the posterior interventricular artery, we can see it goes around to the back of the heart. The left coronary artery is here, and it's going to branch into the circumflex artery and then the anterior interventricular artery. And both of them are going to be coming those first little branches off of that ascending aorta. There are several cardiac veins that you guys need to know that drain the heart muscle. And so we have the great cardiac vein, the middle cardiac vein, and the small cardiac vein. And all of them are going to drain into the coronary sinus, which receives blood from those veins and drains into the right atrium. So if we look here, you can see the great cardiac vein is running down here. Then we've got the middle cardiac vein is going to be in the back, and then the small cardiac vein is going to come here along with that marginal artery. And if we look in the back, you've got that really big coronary sinus. And as I said, if you watch the little model video, it's going to go over the veins and arteries that you guys need to know on the models. Basic circulation routes. We have two different types of routes, the pulmonary circulation and systemic circulation. The pulmonary circulation is going to carry deoxygenated blood from the right side of the heart to the lungs. At the lungs, blood picks up oxygen and releases carbon dioxide, and the blood vessels return to the left side of the heart full of oxygen. So remember with the pulmonary circulation that the colors on the models are going to be switched from what you normally know, because things that carry blood away from the heart, so our arteries, right, and usually they're red, but in this case these arteries carry deoxygenated blood from the right side of the heart to the lungs. And then things that carry blood towards the heart are veins. Usually those are in systemic circulation, we paint those blue, right, because it has deoxygenated heart. But in this case, the blood vessels return with oxygen-rich blood to the left side of the heart. So those pulmonary veins are going to be red on the models. Systemic circulation moves oxygenated blood from the left side of the heart to the systemic cells. So all those arteries are going to be red. At the systemic cells, blood exchange occurs, and then blood vessels or veins return the blood to the right side of the heart, and those veins will be blue. So the basic pattern is the right heart, the lungs, the left heart, the systemic tissues, and then the right heart. And we can see this in this sort of figure 8 diagram right here. And so as you can see, we have our right side of the heart here, where we have, this is our pulmonary trunk, and this is an artery, but it's colored in blue because it has deoxygenated blood. And then these are our pulmonary veins, which are coming back to the atria, and they are going to be colored in red because they have oxygenated blood. So the pulmonary circulation has the colors flipped, so don't forget about that. So make sure you guys know the blood flow through the heart. And I'm not going to read this whole slide, but look at this slide and be able to go through the blood flow. In the heart. One of your worksheets has you drawing it and coloring the arteries and veins. The final thing we'll talk about today is the heart's conduction system. And this initiates and conducts electrical events to ensure proper timing of contractions. And as you guys know, our muscle cells are going to need to have some sort of electrical potential to cause muscle contraction. So specialized cardiac muscle cells have action potentials but don't contract. These are going to be the conduction system of the heart. And so this activity is influenced by the autonomic nervous system. So first we'll talk about the sinoatrial node, or SA node. This is the pacemaker of the heart. It usually initiates the heartbeat. It's located high in the posterior wall of the right atrium. So this is the SA node, and that's where this should depolarize most often, and it's going to be the fastest at depolarizing, and so we'll talk about how exactly that happens in lecture. And this is going to be important for starting the heartbeat. Next, it's going to move through the walls of the atrium and eventually get to the AV node, or the atrioventricular node. After we move to the AV node, the impulse is going to take a pause there, right? The atria are going to contract first. Then we have this nice insulation with that, the fibrous skeleton of the heart. And the AV node is going to conduct that electrical activity down through the AV bundle, or the atrioventricular bundle, into the right and left bundle branches. And then finally, into the Purkinje fibers. And the Purkinje fibers are going to go and spread throughout the ventricle, because the ventricle is so big and thick, both ventricles. And so the spread of the action potential, as we said, it starts at the SA node, goes through the atria. And this is going to be going from one cardiac muscle cell to the other. Remember how the cardiac muscle cells are connected by those intercalated discs that have gap junctions in them, and so the excitation travels through those gap junctions, and the two atria contract together. Next, we have the action potential is delayed at the AV node. AV nodal cells are slower, they have a smaller diameter and fewer gap junctions. Also, that insulation of the fibrous skeleton means there's only one path for the impulse to go through. And this delay allows the ventricles to fill before they contract, so it's really important. Next, the action potential moves through the AV bundle and through the bundle branches to the Purkinje fibers. And then finally, the action potential is going to spread through the ventricles. And so once it gets to the cardiac cells of the ventricles, it can the gap junctions will allow the impulse to spread through all the cardiac muscle fibers, and the cells of the two ventricles contract nearly simultaneously. So we can look at the electrical conduction of the heart by looking at an electrocardiogram, which is called an EKG or an ECG. Skin electrodes are going to be placed on the patient, and they can detect the electrical signals of the cardiac muscle cells. And so EKGs right here, this is what a basic tracing looks like. They have different parts, and I want you guys to be able to identify different parts of the EKG. And so we have the P wave right here, and so that's going to be synonymous with the electrical changes of atrial depolarization, and that originates in the SA node. The PQ segment is going to be associated with when the atria is contracting, or the atrial cells plateau, and we'll talk about the plateau in lecture. And that's going to be this next part here. And then we have the QRS complex. And the QRS complex is going to be important. This has to do with electrical changes associated with the ventricular depolarization. During this time, the atria are repolarizing, but we can't really, we don't see any waves for that because it's blocked by the QRS complex. The ST segment is when the ventricles are contracting. So it's going to be right here, the ST segment. And then finally we have the T wave. And the T wave is going to be the electrical change that happens when the ventricles repolarize. So we can see we've got P is when the atrial depolarize, QRS is when the ventricles depolarize, and then the T wave is the ventricles repolarizing. Next we have a couple of intervals, the PR interval and then the QT interval. And so the PR interval is the time from the beginning of the P wave to the beginning of the QRS. And this is so from when the atria depolarize to the beginning of ventricular depolarization. And so that is the time it takes to transmit the action potential through the entire conduction system. The QT interval is between the beginning of the QRS and the end of the T wave, right? So beginning of the QRS and the end of the T wave. And this reflects the time of the ventricular action potentials, and the length will depend on the heart rate. And we can see this just shows the electrical changes in the atria and the electrical changes in the ventricle, and how that kind of, and we'll look at the depolarization and the plateau, and the repolarization, depolarization and plateau and repolarization that are happening. We'll talk more about this actual action potential and what happens with it in lecture, but that's where those are happening in and how it corresponds to our ECG. All right, so that is all we're talking about in lab today.